Hello dear students, welcome to my channel Kaveri Science Tutorials. Today we are going to see the class 10 Science 2 chapter 1 Heredity and Evolution in that second lecture on the process based on the process protein synthesis. So we will see step by step how proteins are synthesized, how DNA and RNA is involved in the process of protein synthesis, what is mean by protein, how proteins are synthesized. So we will see one by one. We have to revise the structure of DNA and types of RNA before going ahead, and then we will see we have to define protein as a chain of amino acid, and again we have to step by step we will understand the process of protein synthesis, and we have to see the how for the proper functioning of the body of human beings, how proteins are necessary. So in case of human beings, in blood, antibodies, hemoglobin is present in bones, oxygen is present in we can say hair and skin, melanin, keratin. These all types of proteins are present in different types of organs. So in pancreas, insulin is present. So different types of proteins are very much necessary for the proper functioning of the body, and how each and every cell of the body produces protein. So every cell with the help of the cell organelle ribosome. So ribosome is the site of protein synthesis in all living organisms or inside the cell. Ribosome is the place where proteins are synthesized. How we will see one by one. So we will understand the structure of DNA and types of RNA first. So we have seen in the previous standards that the DNA is made up of two strands helically arranged, and these strands are made up of nucleotides. So nucleotide, again it is, I have given the structure of nucleotide here. So nucleotides are made up of pentose sugar, phosphate group, nitrogen bases. In this way, these nucleotides are, there is a chain of nucleotides arranged like this on the strand of DNA, and if the DNA strand is called as 5 dash end and one end is called as 3 dash end, like this 5 dash end and 3 dash end, because there are 5 sugars 1 2 3 4 5. जर तो स्ट्रँड फिफ्थ शुगरला एंड होत असेल 1 2 3 4 5, फिफ्थला एंड होत असेल तर तो झाला 5 डॅश एंड. जर स्ट्रँड थर्ड शुगरला एंड होत असेल 5 पैकी तर तो झाला 3 डॅश एंड. इन दिस वे 3 dash 5 dash are the two ends of strands of the DNA. So nucleotide is made up of pentose sugar, phosphate group and nitrogen bases. Nitrogen bases चे types आपण बघितले, प्युरिन्स असतात, adenine, guanine, pyrimidines असतात, thymine, cytosine, है ना. So means if adenine pairs with thymine, in case of mRNA, adenine pairs with uracil. Instead of thymine, uracil is present in mRNA. And if the guanine pairs with cytosine like this, then the bases are said to be complementary pairing of the bases. Then the pairing is said to be complementary to each other. So complementary means the pairing is proper, then they are said to be complementary to each other. See, there is a process of means we will see the what is mean by proteins first. Important definition can be asked in the board examination. What is mean by protein? Proteins are the chain of amino acids. So for the formation of protein, what we have to form? We have to form the chain of amino acids, then the formation of protein will take place. So here we have to form the chain of amino acids. So here the first definition we have seen, proteins are defined as, if it is asked in the board examination or any examination, you have to define protein as, proteins are defined as the chain of amino acids. So proteins are the chain of amino acids, and the chain of amino acids. So we will define the central dogma of protein synthesis. So we have to see the types of RNA also. So mRNA is also called as messenger RNA. rRNA is the ribosomal RNA, and third one is the means it is the tRNA is the transfer RNA. It carries amino acid and it forms the peptide bond. rRNA forms peptide bond, and mRNA forms acts as a template for the formation of proteins. ठीक है, टेम्पलेट स्ट्रैंड, mRNA strand. So there are two strands of DNA are there, and one strand is after uncoiling of these strands, one strand is used for the process of protein synthesis. So DNA with the help of RNA, all these three RNA are involved. DNA with the help of RNA synthesizes protein, and that process is called as central dogma of protein synthesis. So central dogma means DNA is a genetic material in all living organisms, and there are three types of RNA. Yes, so DNA is present inside the nucleus. So central dogma means the DNA has information for protein synthesis in the form of these nucleotides. This is the information of protein synthesis, and this information is present on DNA. So DNA with the help of RNA produces protein, and that is called as central dogma of protein synthesis. So we have seen the importance of protein for the proper functioning of the human body. Different types of proteins are very much important, so we have to take the proteins in our diet. So how much amount of protein should be consumed, we will see in the after the completion of this important process. Everyone, how much amount of protein should be consumed per day, we will see in the next part of the video. So this is the there are three steps in protein synthesis: transcription, translation, and third one is translocation. So transcription means copy, transcript means to copy. So transcription means the information present on DNA is copied on mRNA, and for the synthesis of mRNA is called as transcription. So in this way, after the process of transcription, synthesis of mRNA takes place, and the mRNA comes in the cytoplasm of the cell. So the first important process of protein synthesis is transcription. Transcription is the copying the information. Transcript means to copy. So the information present on DNA is copied on mRNA, and after for the synthesis of mRNA is called as transcription. So the second process is called as translation. So mRNA has first we will see some important scientist about who has discovered this triplet codon and all that. So mRNA has thousands of triplet codon. Triplet codon is the group of three nucleotide. In this way is called as a triplet codon, and this discovery of triplet codon is done by the scientist called Dr. Hargovind Khurana. So he has got Nobel Prize for the discovery of this group of three nucleotides, that is triplet codon, that can be asked in the board examination, and that is called as the triplet codon. Group of three nucleotide is called as triplet codon, and mRNA has thousands of triplet codon. mRNA synthesized inside the nucleus comes in the cytoplasm because DNA is present in the nucleus. So the synthesis of mRNA takes place in the nucleus. After synthesis, it comes in the cytoplasm, and it joins with the ribosome. We have seen ribosome is the place of protein synthesis. It is the site of protein synthesis. So the mRNA joins with the ribosome cell organelle, and it according to the message present on the mRNA, amino acids are brought by the tRNA, and they are carried to the site of protein synthesis, that is to the ribosome. So in this way, AUG is a group of three nucleotide, and this is the codon which codes for the amino acid methionine. In this way, methionine. So in this way, different triplet codons code for different amino acids, and they are according to the codons or the message present on mRNA. This tRNA brings or supplies amino acids, and these amino acids are joined to each other by the peptide bond. So these amino acids are joined to each other by peptide bond and their chain is formed, and that is called as actual process of protein synthesis takes place, and that is called as translation. So tRNA has anticodons which are complementary to the bases present on mRNA, and this is called tRNA has anticodon complementary to the bases on mRNA, and it brings amino acids according to the bases present on mRNA, according to the message on mRNA, and there is a chain formation takes place. They are joined by peptide bond, and this chain formation of amino acids chain formation takes place. That process is called as translation. So translation is the process of actual formation of protein is called as translation. So this is about the second step of protein synthesis. We have seen transcription, second one is translation, and third is translocation. So translocation means we will see trans means change, and uh, location means third process is translocation. Location means position, translocation. So this ribosome keeps on moving from one end of mRNA by a distance of triplet codon to another end is called as translocation. Trans means change, location means ribosome changes its location from one end of mRNA to another, and that process is called as translocation. So in this way, by the process of translation, such many chains of amino acids are formed, and these many chains of amino acids forms the complex protein. In this way, protein formation takes place by the formation of chain of amino acids. So different according to the requirement of the body, different types of proteins are synthesized by this DNA and RNA, and these proteins are very much important for the appearance of the living organisms and the proper functioning of the body of the living organisms. We can take an example. If there is absence of insulin, the person can suffer from. Insulin is a hormone, insulin is a protein. Hormones, enzymes, all structures inside the human body, they are proteinaceous in nature. So there is a very much importance of protein in our body. Enzymes are also proteinaceous. They are required for the digestion of the food. Hormones, we have taken the example of insulin. Insulin is responsible to reduce the blood sugar level. So if there is absence of insulin, or there is a reduction in the insulin production, then the person can suffer from diabetes. So different hormones are also proteinaceous in nature. Different enzymes required for the digestion, also they are also in proteinaceous in nature. So in this way, we have to take gram of protein as our body weight is there. So not given in the textbook, but we have to take, if our weight is 60 kg, then we have to take 60 grams of protein per day. According to the kg weight, we have to take that much amount of grams of protein. If you are 40 kg, then you have to take 40 grams of protein per day. Per day everyone has to take, if जर तुम्ही 60 kg असाल तर तुम्हाला 60 ग्राम प्रोटीन पर डे कंझ्युम करावं लागेल. कोणत्याही फॉर्म मध्ये, दाल, एग असेल, मीठ असेल, किंवा तुम्ही काय नॉन व्हेज असेल, नॉन व्हेजिटेरियन असाल, व्हेजिटेरियन असाल, त्यानुसार अकॉर्डिंग टू युअर डाएट यू कॅन कंझ्युम ग्रॅम्स ऑफ प्रोटीन अकॉर्डिंग टू युअर वेट. आणि एका पर 1 ग्रॅम per molecule, a gram molecule of 4 kilocalorie energy is obtained from 1 gram of protein. 4 kilocalorie energy is obtained from per gram of protein. So in this way, proteins are necessary for the process of. So we have seen how much amount of protein we have to take per day. Consumption of protein is very much important for the proper functioning of the body. So in this way, we have seen the important processes of protein synthesis, and the whole process I have made it easier for your understanding. And if परत एकदा पॉज करा, परत लिसन करा, परत तुम्ही ही प्रोसेस प्रॉपरली अंडरस्टँड करू शकता. ठीक आहे. दॅट्स ऑल वी हॅव कम्प्लिटेड. याच्यामध्ये तो डॉक्टर हरगोविंद खुराना ऑब्जेक्टिव्ह मध्ये विचारू शकतात एमसीक्यू मध्ये. त्याला नोबेल प्राइज मिळाला ट्रिपलेट कोडॉनची डिस्कव्हरी केली. ट्रान्सक्रिप्शन, ट्रान्सलेशन, ट्रान्सलोकेशन तिन्हीची डेफिनेशन विचारलेली आहे बोर्ड एक्झामला. त्यानंतर इम्पॉर्टंट डेफिनेशन, प्रोटीनची डेफिनेशन सुद्धा याच्यामध्ये विचारली जाऊ शकते. सो proteins are very much we have to consume protein every day. So that's all for today's lecture. I hope you have understood. Okay, so thank you very much. We will continue in the lecture 3 in the next video. Thank you.